concentrations, which makes these concentrations in biofilters and wetlands an interesting area of research. So the millennium drought, as you guys have heard, threatened water security in Australia for over a decade. And the way Melbourne went about addressing this was using stormwater capture, treatment, and harvesting as a new source of water. And also it had the potential to reduce nitrogen and phosphate into the bay. So two treatment approaches were evaluated: constructed wetlands and biofilters. And the reason why you have to evaluate both is that stormwater treatment can be affected by vegetation, filter media, and flow path. And so the fate of nitrate and phosphate concentrations in these systems was evaluated using multiple linear regression.
So our hypothesis was that we could predict nutrient concentrations in constructed wetlands and biofilters by using sensor measurements and other proxy data.
So how did we achieve this? We were lucky enough to visit a total of 6 sites in and around Melbourne, Australia. 2 of these sites were constructed wetlands. This was Hampton Park and Royal Gardens. 2 of these sites were biofilters, known as Hereford Road and Wicks Reserve. One of these sites was a bioretention and constructed pond system, actually the first of its kind in a residential setting. This was Lindbrook Estates. And then we also visited a natural wetland for comparison, and this was the Edith Vale Seaford Natural Wetland. So each site we went to, we sampled at several different locations within the site, but we always made sure we got some data from the inflow and some data from the outflow for comparison. Our sensor data measurements, which we used pH, dissolved oxygen, and temperature, these were all measured with portable instruments that we could lower right into the water and just read the numbers right off the screen. These measurements we took 10 times for each sampling location within the site. And then for the other ones that required lab analysis, we take the water samples in triplicate at the different locations. We brought them all back to the lab, and then we got to bond in the lab while we analyzed them for total suspended solids, chlorophyll, biofilters, nitrate, and phosphate.
And now to the meat and potatoes, or rather the nitrate and phosphate reversion. We use virtual beach basis statistical analysis software from the EPA to develop our MLR models for our data sets. We use nitrate and phosphate as our dependent variables, and these independent variables that you see up on the screen are parameters that we have reason to believe could be correlated to our dependent variables. We have a series of 4 models: the 2 nutrients with wetlands by themselves, and then biofilters and wetlands together. The reason we didn't do biofilters exclusively is because our data set was too small to just consider biofilters. For each of our models, we rank them by the AIC corrected for small sample sizes, and we found that the average R-squared value for phosphate was about 0.6, whereas for nitrate it was much lower at 0.2. However, across all the models, the DO and TSS were the most significant coordinates. Here you have the nutrient concentration on the y-axis and the location on the x-axis. We have 2 plots: biofilters and wetlands on the top, and then just wetlands on the bottom. The light blue line, the thick one for each one, is the observed data set, and then the other 3 lines are the models that we developed. On top, you can see that the models are, while they do explain some variance, there's quite a bit of variance that's unaccounted for. When you exclude the biofilter data, on the bottom you see that the model is a much tighter fit, and the R-squared value here was around 0.65. You see that DO and TSS are the most significant variables.
So why is this important? So we see this high correlation with total suspended solids and phosphate. And we actually know from previous literature that phosphate molecules can easily bind to these suspended solids. And we saw this correlation in our data. So this is strong evidence to remind people that when you're planning constructed wetlands, you need to make sure they achieve sufficient solid settling before the water leaves the wetland. Otherwise, the water is going to be exiting and still taking these high nutrient loads with it unless you get those solids out of the water.
Here we have the same sort of graphs with nitrate concentrations, and you'll notice that the models are much weaker when you exclude biofilter data. Now for phosphate, the model became stronger with the exclusion of biofilters. Here we have, with just wetlands only, the models are pretty much approximated around the average of the nitrogen concentration, the nitrate concentrations. So overall, the nitrate models are fairly meaningless. They don't really tell us much about our data, but we do have a new parameter, phaophyte.
So again, the nitrate models didn't do very much for us. We saw again dissolved oxygen and suspended solids as being important. But we also have the inclusion of this third variable, phaophyte. So what does this say? It doesn't tell us anything really, but it kind of hints at an idea here. We know from our research, we ran a statistical analysis to see how phaophyte and chlorophyll are correlated, and we found that they are. This is because phaophyte is part of decaying plant matter, so there is a phaophyte and chlorophyll relationship. We also know that you can use chlorophyll as a proxy for phytoplankton levels. So here the fact that we see a relationship between nitrate and inverse phaophyte, so one's going up, the other's going down. This kind of suggests that there may be some phytoplankton uptake of the nitrate, and this is a wetland function as well, getting those nutrients out of the water. So we can't say for sure this is happening, but again, when you're designing wetlands, you have to take into account that the phytoplankton can be removing some of the nutrients. Okay, and then we also saw a lot of dissolved oxygen. We saw it coming up as a correlate often. We don't have a good explanation for this, but we suspect it may have something to do with the fact that dissolved oxygen was lower in the shallower areas where there were really high TSS levels.
So looking back at our hypothesis, we can conclude that phosphate can be reasonably estimated using proxy data. DO and TSS did explain a lot of the variability in our models. Nitrate models, on the other hand, did not have the strength. They only revealed a weak relationship with DO, TSS, and the inverse of phaophyte. Future studies with a larger sample size and maybe different parameters may reveal a stronger relationship. Or it is also possible that there is no correlation and that nitrate levels just cannot be estimated using proxy data.
There are references, and we would like to thank the Universities of Melbourne and Monash for hosting us, as well as the PIRE program and the NSF. And special thanks to our advisors and mentors for everything they've done for us these past few weeks. Thank you.
Questions?
I just got something in, and that is, it would be interesting to look at day length on an annual cycle and compare it to phytoplankton populations and those parameters you looked at.
Yeah, that's true, because we definitely noticed when we were there, it would get dark around 5 o'clock because we
Yeah.
were there in the winter. So we could go back.
So they're saying they should stay there. Any other questions or comments? Excellent job, guys.